Cellular respiration is the controlled release of energy from organic compound in cell to produce ATP. Here, the controlled release means metabolic pathways and cycles controlled by enzymes, and organic compounds are usually glucose or carbohydrates and sometimes lipids and proteins. Here, the cell refers to all organisms respire for cellular activities, and ATP here releases energy by breaking the high energy bond. Cellular respiration can be anaerobic or aerobic, but the general equation is And cellular respiration is divided into largely four processes: glycolysis, link action, Krebs cycle, and electron transport chain with chemosis. So first, in glycolysis, glycolysis occurs in the cytoplasm and does not require oxygen. And here, glucose, or a hexose sugar, is phosphorylated into hexose biphosphate through using 2-ATP. And then this hexose biphosphate is split into 2 triose phosphate. And this process is called lysis. And through ATP formation and oxidation, 2 triose phosphate turn into 2G3P. Here we oxidize NAD+, and then make 2-ATP. And finally, the 2G3P is converted into 2-pyruvate by another ATP formation. So in total, we say because 2-ATP is consumed and 4-ATP is produced, the net gain of ATP in glycolysis is 2-ATP. After glycolysis is link action. Link action, by general definition, is a process that cut carbon chain into usable forms. And the pyruvate that was made in previous step, it undergoes oxidation and decarboxylation. Into acetyl group. We oxidize 1 NAD plus into NADH, and 1 CO2 is released. And this acetyl group is combined with coenzyme A to produce acetyl-CoA. And because there was 2 pyruvates, the both pyruvate undergoes the same link reaction and produce 2-acetyl-CoA. Here, this link reaction can occur to fatty acid. However, the link action of fatty acid will not include the decarboxylation part. Third is the Krebs cycle. Here, the acetyl-CoA is 2-carbon molecule, enters the Krebs cycle in the matrix. And this 2-carbon joins with 4-carbon sugar called oxaloacetate to produce a 6-carbon molecule called citric acid. And citric acid 6-carbon molecule undergoes oxidation and decarboxylation to produce a 5-carbon molecule by converting NAD plus into NADH and releasing 1 CO2. C5 molecule undergoes another round of oxidation and decarboxylation to produce a C4 with NAD plus converting to NADH and also releasing 1 CO2. Here, this C4 is not the final form of C4 oxaloacetate that joins with acetyl-CoA to produce citric acid. So we have to go through rearrangement. And in this rearrangement, with addition to rearrangement, it also undergoes phosphorylation. So in rearrangement, NAD plus is converted to NADH. FAD is converted to FADH2. And then also 1 ATP is produced. Finally, we have electrotransport chain and Krebs chemiosis. So chemiosis is the diffusion of ions across a semi-permeable membrane through a carrier protein, and the high concentration of H plus creates this electrochemical gradient, and because of this, H plus moves through ATP synthase by chemiosis, which drives the rotation of ATP synthase, and ATP is produced. So in this electron transport chain, there exists an ATP synthase, this part is intermembrane space of mitochondria. This is the membrane, inner membrane, and then this part is the matrix of mitochondria. The electron carriers such as NADH and FADH2 that was created before deposit their electrons and hydrogen ions, and a series of integrated proteins, integral protein complexes located in the membrane act as the electron transport chain, and protein electron carriers include FMN, FES, Q, cytochrome B, C1, C, A, and A3. And as they drop off the electron, the high energy electrons passes from carrier to carrier, and the energy of this electron is used to transfer H plus across the inner membrane from the matrix to the intermembrane space. So the hydrogen ions here are transported to the intermembrane space. And in order to allow the electron to continue to flow, they must be transferred to a terminal electron acceptor at the end of the chain. And this is done by oxygen, and it forms water. And as I said before, as these electrochemical gradient is built because of the high energy electrons, these H plus will like to diffuse back. And in order to diffuse back, they move through this ATP synthase. And when this ATP synthase, H plus move back through this ATP synthase, the ATP synthase turns and an ATP is released. This is the aerobic cellular respiration. And to go a little bit deeper, there are differences between aerobic and anaerobic cellular respirations. Firstly, they're similar in a way that they both start with glucose and both produce pyruvate by glycolysis because glycolysis does not require oxygen. And also, of course, they both produce ATP. However, the differences is that for aerobic, it requires oxygen, such as this chemiosmosis, and then it also has a high yield of ATP, while an anaerobic reaction has a low yield of ATP because it does not contain the electron transport chain. And then for the waste products, the aerobic respiration produces a waste product of CO2 as well as H2O, while anaerobic have CO2 and ethanol as waste product for human, and then for bacteria, and CO2 and lactic acid for human. For aerobic, they can metabolize other molecules like fatty acid mentioned before, and for anaerobic, it does not require mitochondria and occurs in the cytoplasm only.